Hello, everyone, and welcome to today's webinar, Studying Protein Polymerization with Mass Photometry. I am Morgan Sterling of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more, visit refine.com. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits after the webinar. We do encourage you to participate in today's event by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Nikolas Hundt, Postdoctoral Research Associate, Ludwig, Maximilian University of Munich. Nikolas, you may now begin your presentation.
Yeah, thanks for the kind introduction. I would like to thank Refine at this point for sponsoring this webinar. And I would like to thank all of you for your interest in my webinar. I would say let's jump right into it. You've all probably heard in one or the other way about these proteins that I've listed here. They are all biopolymers, which are involved in many different important cellular processes. For example, up there you see actin and microtubules, which are an essential part of the cytoskeleton, and they are basically involved in all the processes where cells need to change their shape or move. For example, for production of cell protrusions or cell migration, cell division, and so on. And then there's another group of common biopolymers, which are called amyloids. They are actually not one type of protein, but many different ones. And amyloids are rather a structural state of proteins, where proteins form polymeric beta sheets, and then from this over time form three-dimensional structures, which are called fibrils. And they are important for a number of very prominent disorders. They're usually associated with the progression of the disease. The examples that I've listed here are, for example, important for Alzheimer's, Parkinson's, Huntington's disease, diabetes, and Creutzfeldt-Jakob's disease. In order to understand the regulation of all these processes, it's of course crucial to understand how these polymers are formed. And how do you actually study the assembly of a biopolymer? Well, you can start off by understanding what your polymer looks like structurally, for example. For this, you can employ high-resolution techniques such as X-ray crystallography or cryo-electron microscopy. And this way you can, for example, obtain atomic structures of your monomeric subunits, as well as the polymerized state of your protein. But of course, looking at only the structure will not tell you much about how the polymerization process is regulated and how it actually functions dynamically. So if you want to learn something about the polymerization process dynamically, you will probably go to a less resolution technique. Usually light microscopy techniques are employed for this. Very common is fluorescence microscopy, where you label some individual subunits of your polymer with a fluorescent dye, and then you can watch individual polymers grow and shrink over time. This is very useful to get like basic kinetic constants for the growth and shrinkage out, as well as to understand how, for example, regulators influence this process. But For the regulation, it's not only interesting or important to look at the overall polymerization process, but it's also important to understand how the polymerization process starts. And there, it becomes a lot more difficult to assess this initial part of the assembly, because we cannot employ so easily these optical techniques anymore. And up until now, it's actually common to explore this process just with bulk techniques. And typically, a bulk polymerization assay uses different kinds of bulk signals, such as fluorescence, scattering, viscosity, or birefringence measurements, to follow the accumulation of the polymer over time in solution. And this polymerization signal can usually be subdivided in 3 different phases. The first one is called the nucleation phase, where the line stays mostly flat. This is where the polymerization is usually started. Then you go into the elongation phase, where the signal will rise much more quickly. And this is where the major portion of the polymers is formed. And then later on, you enter a stationary phase, where polymer growth comes to an end and enters more like a steady state, where growth and shrinkage is more or less in equilibrium. Now, from these polymerization curves, especially if you do them at different polymer concentrations, you can deduce somewhat rates for the initial steps, as well as an overall polymerization rate. But as you might guess, since this nucleation phase actually just happens in this flat regime of the curve, there's not much information really about the molecular pathway that underlies this polymerization process. And usually you have to come up with a kinetic model and try to simulate your bulk polymerization curves based on that kinetic model. And this is how we figure out different molecular pathways during the nucleation process. For actin, the first models have come up in the 1960s and 70s. And these nucleation models have been refined over the decades up until 2001, where I think, in my opinion, the most detailed model came out for actin polymer nucleation. David Sept and Andrew McCammon tried to simulate all possible nucleation pathways for actin based on a Brownian dynamic simulation. And essentially what they did was, as you can see on the left, the actin polymer consists of 2 helical strands, the blue one and the red one. And each actin subunit can have contacts to the neighboring longitudinal subunits in the same strand, as well as a cross-filament contact with the subunits on the other strand, basically. And then they've essentially simulated how stable all the possible combinations of nuclei were based on their Brownian dynamics, and figured out a major pathway in which this nucleation process should happen, which is encircled with a dashed line here. So basically you can have 2 different dimers, was their conclusion, which are relevant. One is the longitudinal dimer and the cross-filament dimer. And then they figured out growth, like association and dissociation rates, as well as dissociation constants for each of these steps. And their major conclusion was that the rate limiting step is the formation of a dimer. And once the system has reached a trimeric state, afterwards, all the subsequent steps are more or less working in the same way with the same rate constants. And then they use this model and scaled their theoretical rate constants to experimentally determined ones and compared their predicted bulk scattering curves with experimental ones, and found that their model would explain it quite well. And they could even determine time courses for the individual different nucleation species over time. But unfortunately, up until now, this theoretical model could not be confirmed experimentally. And you would ask yourself, why is that the case, that after more than 20 years, it was not possible to reproduce this experimentally? Well, how would you measure this? Essentially, the simple task would be to quantify all polymer species dynamically using single molecule imaging. Well, yeah, I mean, who has ever tried to quantify this experimentally, for example, with single molecule fluorescence imaging, knows that this is a pretty hard task to do, especially if there are more species than just a couple. But fortunately, more recently, we introduced a new microscopy technique called mass photometry, which is designed to do exactly that task. And since you're sitting in this webinar, you're probably familiar with the technique, but still I will go through the basic principle very quickly. So you shine a laser onto a cover slip, which holds a droplet of your protein solution, and then you record the light that comes back onto a camera, and the light that comes back in this case is a combination of the reflected light from the water glass interface and the scattered light from little objects that are sitting on that interface. And the reflected light creates a bright background, and wherever a particle is sitting on the surface, the scattered light will generate a small destructive interference, and hence you will see kind of a dark dip at that position. And the darkness of that dip is directly proportional to the size of your particle that you're looking at. And you can calibrate that signal using proteins of known size. And since all the proteins have very similar optical properties, you can basically get a mass distribution of your proteins that land on the glass cover slip. And I've done this for Actin, for example, and this is a mass photometry video that shows Actin molecules landing on a glass cover slip. So every time you see a dark dot, that's a landing event. And the reason why they appear and disappear is just because we continuously remove the static background. So once a particle has landed, it becomes static on the background and gets removed by the image processing algorithm. This way we can image all the proteins that are coming in one after the other and analyze their darkness, basically. And then when you plot the darknesses or the masses of all your molecules that you have in your video in a histogram, you can generate a size distribution of the protein and get an impression of what type of molecules and how many are present in solution. And this is very powerful because the mass resolution with mass photometry is so high that we can actually distinguish the different oligomeric states as individual mass peaks in the size distribution. And now this is also very useful for the quantification of the initial polymerization reaction, since this is exactly what we needed, what I talked about before. And you can also do that. As a timed experiment. And the way I've done it, I've taken a monomeric actin solution, added the polymerization starters, let the polymerization incubate for a while, and after different time points, I pipetted some sample out of that polymerizing solution into a cover slip with a droplet of buffer, and immediately watched landing of the molecules that had formed after these different time points. And if you then compare the mass distributions of different actin concentrations at different time points, you will see that if you're working at or below the critical concentration of filament formation, which is the red curve, there's not a significant difference at the different time points. But if you work above the critical concentration, the equilibrium shifts to larger molecular masses. And this can be crucially quantified by taking the area under the curve of this histogram. And as you can see, as I said before, like beyond the critical concentration, you get growth essentially, and below the critical concentration, the mass distribution stays more or less constant. But this is really just a very crude analysis. The actual advantage of this lies in the fact that we can quantify principally every individual species that is present during the polymerization process individually, and take a time course of it. And the way I've done it, I took the size distribution and used a combination of Gaussian fitting plus for the well-defined peaks, and just simple binning for the less well-defined peaks for the higher masses, and determined molecule counts for each oligomer size. And this can, since I know the overall concentration of actin, can be directly converted into concentrations of each of these species. And from the Gaussian fit, I also know some parameters from the experiment. So in principle, from any of these oligomer size distributions and counts, I can recreate based on the fit position of the peaks, as well as the width of the peaks, which corresponds to the mass noise. Basically, I can regenerate a simulated mass distribution, which I can directly compare with my experimental histogram. And this is all I need to essentially now test all the models that are out there and compare them with my mass photometry measurements. So I took the Sept-McCammon model and simplified it a little bit by just breaking it down to the main pathway, because all the other pathways are so unlikely that they don't really contribute to the overall kinetics. So essentially, I took the formation of the longitudinal dimer, and then from there the pathway to the trimer, tetramer, and all the subsequent steps, and use the rate constants that are provided in this paper. And then I needed to solve a couple of, derive a couple of differential equations to describe this equilibrium, basically in order to be able to create time courses for the concentration of each individual species. The only assumptions I made was I had to limit the number of possible species to an infinite number, which in my case was 400, because otherwise I couldn't compute this. and I had made the assumption that every step would just be by the addition of a monomer and not bigger species. And then one last thing I needed was a starting distribution for which I took the experimentally determined one at 1 minute. And then I let my simulation run. And from the concentration time course, I could pick the 5 minutes, 10 minutes, and 15 minutes time point, like I did in my experiment, and recreated from my concentration time course an experimental histogram that I could compare with my mass photometry data. And as you can see here, the Sepp-McCammon model predicts overall net growth over the course of the 15 minutes to masses up to 7 megadalton. But in the low mass regime, it actually predicts almost no abundance of dimers and trimers because they're incredibly unstable according to their model. But if you remember my experimental data, there were actually quite significant amounts of these small oligomers. So the simulated traces didn't match at all my experimental mass photometry data. I can do the same. I can use the same simulation to also produce artificial bulk scattering curves, which I did, and compared it to an experimental measurement. And here I actually kind of could recreate a very similar curve, which means that the Sepp-McCammon model explains the data in the bulk, but it could not explain my mass photometry data. So my first attempt to overcome this problem, I tried to adjust the rate constants of the model until my experimental data and the size distribution perfectly matched. But then I had the issue that I didn't observe any net growth into long filaments, as you can see in the top time course of the distributions, and also there was the light scattering curve was just a flat line basically. This was a bit puzzling because that meant that the common model could not explain at the same time my bulk data and the mass photometry data. So I stepped back and thought again about what these nucleation models were considering, and I noticed that they weren't really considering all the information that is out there about actin. So first of all, actin is a polar polymer, which means that one end is different from the other end, and the different ends have different association and dissociation rate constants. This is something that these models haven't considered so far. And also, we know that actin is an ATPase, which means usually ATP actin gets preferentially incorporated at the barbed end of the filament. And then once it's incorporated, it can cleave ATP into ADP and phosphate, and then later on release the phosphate. And the ADP actin then preferentially leaves the filament through the pointed end. And the common nucleation models completely disregarded this fact so far. So I thought, okay, maybe I can modify my model a little bit and produce a simple model where I have one equilibrium where the filament can grow from both ends. And then I noticed that ADP-PI actin has has a really slowed down dissociation rate at the pointed end, which means that essentially if the final subunit at the pointed end hydrolyzes its ATP, it will stop depolymerizing from that end. So essentially I have a slow transition from this top equilibrium to the bottom equilibrium with different rate constants. When I plugged this into my simulation, I actually got very different curves out. And in fact, just using published rate constants for the polymerization and depolymerization rates from the barbed and pointed ends already almost perfectly recreated my experimental data in the low mass regime. Plus, I got net growth and could actually describe the bulk scattering curves much better than before. And to confirm, I used the same kinetic rate constants and simulated data for different actin concentrations. And in all the cases, I could explain my experimental data fairly well with this new model. So I think this worked pretty well, but there was still a drawback. And actually this came especially during the review of the paper. In fact, if ATP hydrolysis is the actual trigger of actin polymerization, how come that actin can still polymerize without ATP hydrolysis? For example, you can replace ATP with a non-hydrolyzable ATP homolog, AMP-PNP, and you would still get filaments. And also you can polymerize actin just from ADP actin. And there have been papers that you can polymerize actin with no nucleotide at all. So, okay, how can we explain this? In fact, actin can also bind, usually for polymerization, actin needs to bind divalent cations. And in order to keep it monomeric, actin is usually kept in a calcium solution. And then for polymerization, you exchange the calcium for magnesium. And calcium actin has different polymerization rate constants than magnesium actin. So in fact, I tested what happens if I do a mass photometry experiment with AMP-PNP actin, and then use the rate constants for calcium actin and magnesium actin and the exchange rate with the same kind of model on that data. And in fact, I could explain the data that I got out again very well. And I even observed that ATP actin would give me a lower amplitude in my bulk polymerization curves than the AMP-PNP actin. And the simulation with this model could even explain the discrepancy in the amplitudes in these two different experiments. Yeah, so now finally, you might ask yourself, why do the results in the bulk of the nucleation models and these new completely different models actually look so similar? Well, in fact, this is mostly because the two principles, the kinetic principles, are almost the same, in my opinion. For the nucleation model, you have one equilibrium with a less favorable polymerization, which is described by a steeper decay in the size distribution at the beginning of the size distribution, and then a slow transition to a second equilibrium, which with much more favorable growth rates, which can be explained by the other half of the and the other slope of the size distribution. In the new model, it's basically the same. You have one equilibrium, which is explained by the first half of the size distribution, and a higher slope in the decay of the species. and a slow transition to a second equilibrium, which is more favorable for growth. And I think this principle is the reason for the shape of these bulk polymerization curves. But of course, on a molecular level, they work with two completely different pathways. And you can even and we even identified two different pathways for actin. One was the calcium to magnesium transition. If you look at it from a more thermodynamic point of view, you can say the calcium state has a less favorable Gibbs free energy of polymerization, and the magnesium state has a more favorable one. And then in pathway 2, actin changes its Gibbs free energy of polymerization to a more favorable one by ATP hydrolysis. And in both cases, you can generate net elongation of actin filaments. Well, I do think that this doesn't have to stop just at actin. As I mentioned earlier, there are these amyloid fibrils, which are very important for the generation of different diseases. And in particular, nowadays it's thought that the onset of these polymerization reactions are the actual cytotoxic events that are relevant for the progression of the disease. And therefore, since these systems lack the same methodology for investigating the small oligomers, I think mass photometry can really help to gain more insights into these disease mechanisms. Finally, I would like to thank a couple of people, especially my previous lab mates from the Kokura lab in Oxford. They are an amazing group of people, and they are really good microscope builders. Without them, it would have never been possible to image and do mass photometry on actin at this quality. I would also like to thank my collaborators from Oxford who helped me a lot in the data analysis, and also Josiah Kane from Refine who helped me with data analysis, and our collaborators from the NIH, Jim Sellers and Harry Takagi, who provided me with the raw material for producing actin. And finally, I would like to thank my new supervisor, Claudia Feigl, here in Munich, who gives me the opportunity to explore amazing new avenues for applying mass photometry. And finally, I would like to thank you for your attention.
Thank you, Nikolas, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the far left of your screen. We'll answer as many questions as we have time for. So the first question that came up, Nikolas, was what software that used for analyze nucleation model, and is that free?
So I produced the analysis software myself. I mean, for producing the mass distributions, of course, I just used the normal refine software. Sorry, I need to go on that side. I used the normal refine software for creating the mass distributions, but the software for analyzing and simulating the mass distributions I created myself in a Python package, and it is freely available. If you check out my publication in Science Advances, then you will find a link to a repository, and I put all the code on there so you can download it if you like.
Great. And is time travel possible using mass spectrometry?
I don't think so. Not yet.
Great. And then we have a bit of a situation here. So someone said that they used MP to study Huntington fusion protein, but while the protein had a nominal mass of 58 kDa, using the collaboration curve, it came out to be calculated as 74 kDa, which is significantly different than a predicted 5% error. Can this difference be attributed by the expanded glutamines or something else that isn't clear?
Good question. From just this, it's hard to tell, I must say. Are you sure there was nothing else in the sample? I mean, it could be if there is if you co-purify a binding partner for the Huntington fusion protein, maybe it sticks to the protein or something, or maybe a chaperone or something that because if you would detect a complex, that could explain why you get a mass increase. I don't know, if you like, you can get in touch with me. Maybe we can have a look at the data together, and I might help you resolve that issue.
Next question. Excellent work, Nikolas. Would you be able to tell how well MP is compatible with high 300 mm NaCl concentrations?
Oh yeah, so that's not a problem actually. I've used even higher sodium chloride concentrations. The only difference for higher salt concentration is that the buffer then has a different refractive index than at lower salt concentrations. So if you do your mass calibration, you should always use the same buffer and same salt concentration as you use for your protein of interest. Because the slope of the mass calibration might be slightly different at these high salt concentrations.
Great. And how did mass photometry help you resolve biases with kinetic model overfitting?
Yeah, so I think the main advantage of mass photometry for for these polymerization reactions is really that you can quantify the amount of small species that are present during the polymerization process, and that way it's a lot easier to find a molecular pathway for the reaction than just using bulk methods.
Awesome. And Nikolas, are there any drawbacks to, excuse me, using mass photometry compared to classical bulk-based methods?
Yeah. Yes, I would say there is, because I mean, most actin experiments so far have been done at much higher concentrations. As you might have noticed, the concentration that I've been working with were already quite high for mass photometry. But I had to, because the critical concentration of actin is 120 nanomolar, and that's already fairly high. But in order to see something, I had to go higher than this. And the way I overcome this problem in my case was that I put a droplet of buffer onto my cover slip beforehand, and then I diluted my actin solution into that droplet and hoped that the complexes that were forming didn't fall apart during the time of the acquisition. That's how I had to do it in this case. So I think one of the major drawbacks for these kind of polymerization reactions is really that you might not get to the concentrations that you need for polymer formation.
Great. And it looks like we have one more question. If anyone has any more questions, go ahead and ask them now. The next question for you, Nikolas, is how does mass photometry compare to other single molecule techniques for the study of protein polymerization? Did you consider using any of these other techniques?
Not in particular for the actin polymerization. I think the reason why there has never been really an experimental study, single molecule study on actin nucleation is because it's super difficult with other techniques. For example, if you would be using something like single molecule fluorescence and do something like stepwise photo bleaching and so on, we would probably be able to detect the dimers and trimers that we've seen with mass photometry, but it is much harder to quantify these, and especially for this huge number of different species, I think it's really hard with especially fluorescence techniques to get the same quality of data that we got with mass photometry.
Awesome. Great. Well, looks like that wraps up our questions. Thank you, Nikolas, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Refine, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for, or if something else comes up, in the on-demand period, we will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it is available for replay. We do encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you, Nikolas, and goodbye.
Thank you. Goodbye.